Good day, everyone. It's my honor and pleasure to be able to talk to you today about precision medicine, which, as evidenced by my recent email activity, is a very important and current topic. And a good place to start, I thought, would be to try to define exactly what precision medicine is, given the frequency with which we're hearing about it now. So if we go to perhaps one of the first definitions, this was from the National Research Council in 2011. And per the National Research Council, precision medicine was defined as tailoring of disease prevention and treatment based on the characteristics of each individual. So throughout this presentation today, for the next 45 minutes or so, one of the points I think that I will be making is that this is an old idea, but it has new implications as a result of extraordinary new technology that's been developed and our ability now to do large-scale genomic data analysis as a result of these modern technologies. As a secondary definition, President Obama developed and issued his Precision Medicine Initiative in 2015. And in the President's initiative, he defines precision medicine as an innovative approach to disease prevention and treatment that takes into account individual differences in people's genes, environments, and lifestyles. So perhaps much more comprehensive, and again, focusing on genes and the genome, and we'll talk more about that over the course of the next 45 minutes. As a part of President Obama's Precision Medicine Initiative, he has requested $216 million in the 2016 budget, and he has 5 goals proposed to develop this initiative. The first goal is to develop more and better treatments for cancer. Secondly, to create a national voluntary research cohort. Third, to develop better mechanisms to protect privacy given the genetic data that's going to be as a part of this initiative. Fourth, to modernize regulation, as we'll talk about later with some of these novel study designs, the regulation that will need to accompany it. And finally, to develop public and private partnerships to move this initiative forward. So if you look at the core premise of precision medicine that these various definitions and initiatives are trying to achieve, Aronson suggested that one of the core premises, the application of evidence-based medicine can improve quality and control costs in the healthcare system. And he underscores the fact that the public's current perception of precision medicine and its core premise is probably overly simplistic. That is, the current perception is that a patient will have a test performed, such as sequencing the genome of their tumor. Based upon that, the physician will look at that information and identify an actionable target. And actionable we'll talk about more in a moment, but it really means a target for which there is a drug or a therapy available, whether that's an approved drug or therapy or an experimental drug or therapy. So they look at the test results, they pick a target, an abnormality for which there's a drug, and they treat the patient with that particular drug. So while that's a very nice simplistic construct, Aronson goes on to suggest it's probably overly simplistic. And just to highlight a few of the additional steps that are not a part of this overly simplistic process, we likely will have to consider the diagnostic test performance. And so it's a subject of a different presentation, but companion diagnostics, the ability to do these tests and identify these abnormalities in meaningful ways, much more complex than we currently imagine. There's the complexity of interpreting the test result. With cancer, there are likely to be multiple targets as opposed to one single target. How then are those targets prioritized, and what are the driver mutations and driver abnormalities that are required for the cancer to arise and survive, as opposed to all the other abnormalities that are there that aren't driver mutations or abnormalities and are just along for the ride, if you will. Then there's the ability, the need to pick targets that are actionable, where we have an approved or an experimental medication that might actually target that abnormality or abnormalities. And then you have to have the availability of those experimental or approved drugs. Those drugs or treatments need to have been tested in patients with specific abnormalities and proven to be effective, and that the safety and efficacy of those, especially if used in combination, has to be known. There's the enormous complexity around clinical decision-making and management. There's the complexity of healthcare delivery variables and access to not only the test, but the potential drugs and therapies. And then how do you measure meaningful outcomes as a result of this? So as you can see, as Aronson suggests, the current simplistic interpretation of what precision medicine is, do a test, pick a drug, treat the patient, is probably very, very simple and naive given just a small listing of other factors that are likely involved. So with that understanding of the definition and the core premise, the goals of precision medicine then would be to determine how can we better target driver or central oncogenic events, the abnormalities in patient tumors. How do we determine who will respond to treatment? Conversely, how do we identify who will not respond to any given treatment? We also, as physicians, need to understand that, especially if the treatment is not terribly effective, how do we prevent or reduce harm with the therapies that we pick? How can we make these treatments more cost-effective? How can we improve access to these more meaningful treatments to get better treatment outcomes? And since so much of precision medicine is focused on the human genome, will molecular testing actually improve outcomes by targeting the genome? Switching gears just for a moment, because you often hear precision medicine and personalized medicine used in the same sentences and in the same context. So perhaps a comment about personalized medicine. So just recently, Lippi and Plebani stated, as it relates to personalized medicine, that although the term personalized medicine is repeated like a mantra in scientific papers, meetings, media, and even the internet, its exact definition is vague and remains almost unclear to many. So unlike precision medicine, where we have at least the Obama administration's definition and others, personalized medicine is probably a little less well-defined. So the question that I'm often asked is, are personalized medicine and precision medicine the same? And the answer is probably not clear, but the terms are used interchangeably quite often. And I would suggest thinking about these in two different arenas. First, the clinical care setting as opposed to the clinical research setting. So take the clinical care setting first. I think appropriately and respectfully to physicians who care for patients and have cared for patients over the past many centuries, physicians always try to personalize their care of the patient, and physicians always try to be as precise with the treatments that they deliver as possible. That's perhaps very different than the research environment, where the definitions of personalized and precision medicine are perhaps a little different. And so, as we know, in the clinical research setting, what we learn from the individual patient tends to be very limited, and so we tend to have to aggregate patients into groups based upon certain characteristics to be able to ask and answer questions in a meaningful way. And the relevance of that to the specific individual may or may not be important, and that's one of the large challenges of clinical research. So clinical care is increasingly precise and increasingly personal as technology advances and as knowledge advances. So I would suggest that instead of using the terms precision medicine or personalized medicine, because these are processes, not a final result, these are a journey, not a destination, perhaps a modifier to the terms such as increasingly precise medicine and increasingly personalized medicine perhaps may be more appropriate, and that is probably not only more appropriate, perhaps more respectful of physicians who do their very best to deliver personalized and precise medicine to their patients. So again, the concept here, perhaps one of the things to remember is that this is a process, it's an approach, and not necessarily an end result or destination. So because of the the amount we hear about personalized precision medicine, I ask myself these questions. Is this really new terminology? Is this a fad? Is this political? Is this marketing? Is this media? And I think the answer is probably yes to all of the above. That doesn't mean that it's not important, but we clearly are at a point in time where these thoughts, these processes, these approaches have come to the fore as a result of remarkable advances in biology and technology. But that said, it is a hot topic at the moment for all these reasons. I would also argue, as a pediatric hematologist oncologist who's had the exceedingly rare privilege of working in both the pediatric and adult cancer environments and cultures throughout my career, the strategy isn't new. And so these approaches to increasingly personalize and be increasingly precise in the therapy we deliver to cancer patients has been exactly what has been happening over the past 60 years. So one of the examples of this, one of the great success stories in all of medicine, is the treatment of childhood leukemia that started with Dr. Sidney Farber and others in the 1940s and 1950s. So if we look at the 10-year survival estimates for children who have acute lymphoblastic leukemia going back to the late 1960s, we see that the 10-year survival has increased from less than 10% to over 80% right now. And based upon certain risk groups, which we can talk about in just a moment, in the very good risk groups, survival and cure now exceeds 90 and 95% in some of these subgroups. So remarkable progress in less than 60 years for the outcome of these children with this terrible disease. So how do we achieve this remarkable improvements in outcome for children with ALL? The large answer to this, the predominant answer to this, is that clinical trials have been increasingly precise and personalized since Dr. Farber and others worked in the 1950s, and this has been based upon an increasing knowledge of biology. Biology, biology, biology. And based upon biology and other factors, the ability to identify risk groups, and as a result of risk groups, to identify risk group-directed therapy. This is personalized because of a child's individual risk group, and it's becoming increasingly precise as we understand more of the biology and it can improve our risk groups and hopefully at some point the therapies for those risk groups. And just as one illustration, this is a pie chart from Charles Mulligan just identifying some of the cytogenetics of pediatric ALL. You can see the large number of abnormalities, many of which are now actionable or targetable. And just this one aspect of the disease is increasingly complex. And you can see as we've learned more about the biology, the improvements have followed. So as a tangent, but an important tangent as it relates to precision medicine, the question is, does an improvement in outcome require new and expensive approaches? Again, coming back to our biases that precision medicine is predicated upon new, very expensive, very complex therapies. And that may be true, but not necessarily. So I would just want to make sure that you understand one of the points that increasingly precise medicine may be based upon using known drugs and known technologies and treatments and learning how to better use them. So on this particular slide, you can see when the FDA approved the drugs that we commonly use today to treat children with acute lymphoblastic leukemia. So we've used pretty much the same group of drugs since the 1950s. Methotrexate was approved by the FDA in 1958, still a mainstay of the treatment of children with ALL. The new drug on the block is asparaginase, which was approved in 1994. So if we look at the outcome from children who have ALL, the slide I showed you a moment ago with that remarkable increase, that increase is the result of using the same drugs but learning how to use them, optimally using them. So increasingly personal and increasingly precise medicine can be the result of learning how to use known drugs better and optimally, and it isn't necessarily dependent upon a new, expensive, biologically targeted drug. And as a result of this, this is data from SEER that was published by Malcolm Smith, Greg Riemann, and others, the leadership of CTEP and the Children's Oncology Group a couple of years ago, where you see the mortality rate for childhood cancers. The bottom curve in the yellow is leukemia lymphoma and reflects the remarkable improvement I was just showing you for childhood acute lymphoblastic leukemia. The upper blue curve are solid tumors. And you can see gratifyingly since the 1970s and mid-70s, the mortality rate has fallen. And we continue to make improvements, particularly for leukemia lymphoma, by optimally using largely older drugs. But you can see for solid tumors, the curve began to plateau. The mortality began to plateau in the mid-1980s. And the slope of the curve for leukemia lymphoma, the yellow one, seems to be tapering off a bit. So what this would suggest is that progress is slowing. For almost 60 years, we've optimized therapy with the current medications we have. And so now to be able to continue to improve the outcome and get off those plateaus that we're seeing for some of these diseases, what do we do next? So that said, over the past 60-plus years, effective cancer has been based upon 4 foundational approaches and modalities of therapy. We treat cancer with surgery, radiation, conventional chemotherapy, and blood and marrow transplantation. And we've made huge progress using these 4 modalities of therapy. And there's undoubtedly progress that continue to be made using these particular areas. But the plateaus that we saw from the previous slide says that we're plateauing, and so these 4 modalities of therapy, the possibility for improvement may be increasingly limited. So to make further improvements, we're going to have to add something new, and these treatments will undoubtedly be based upon biology of the cancer and the patient, the host. And one example of that, which is evidenced by many of the precision medicine projects, is going to be based upon sequencing the genome. So, how many genes are involved in cancer? So current data would suggest that we as humans have approximately 20,000 genes that allow us to be human. Of those 20,000 genes, it's fairly well established that based on current knowledge, only about 1% of all of our genes are involved in cancer and mutation, and only about 1% of genes are involved in the cancer process. And of those 1% of genes, roughly about 300 or so, we have a relatively small group that are currently actionable, meaning we have a drug that can target that particular abnormality. And so that number is probably several dozen that we have actionable targets based on what we currently know about the abnormalities in these genes that are part of cancer. And we've taken great advantage of some of those abnormalities already, which have resulted in great excitement about precision medicine. And so some of the examples, probably the seminal work that really created this whole thought process was the work by Brian Drucker and others with imatinib and BCR-ABL CML. And then I could cite many examples, but some that are particularly exciting are work with EGFR mutant lung cancer and then with HER2-positive breast cancer that you cited here. And there are many others. But as a result of this approach, already some significant advances are already being seen. So the process to date, as I alluded to earlier, is that we identify genomic alterations in patients who have conventionally diagnosed cancer, typically based upon histology or an organ of origin. The genomic alteration or the driver, the central mutation, is identified, and this is based upon some exceedingly complex and elegant and beautiful biology. And then the activity of a target drug has been tested in the preclinical and early phase clinical work, much like happened with CML and EGFR mutant lung cancer and HER2-positive breast cancer, et cetera. So one central question to precision medicine is the same approach effective regardless of the patient's conventionally defined diagnosis? So what is the current landscape and what is happening out there? So Roper and colleagues this year published a study where they looked at adult interventional cancer trials registered in clinicaltrials.gov, and they looked at trials between September of 2005 and May of 2013. And a trial was defined for this particular study as precision cancer medicine if the genomic alteration was in a predefined subset of 88 genes that were required for enrollment on these particular studies, coming back to the concept there's a relatively small number of genes we know to be involved in cancer. They identified almost 19,000 trials, of which 48% were eligible for inclusion in the study based upon their definitions. And of these, 8% were classified as precision cancer medicine trials. This resulted in an increase of 3% in 2006 to 16% in 2013, again showing the advances we're making, the interest, and the likely benefits as a result of this. So the number of trials is increasing. The trials that were there were most often in breast cancer, colorectal tumors, and skin cancers. And interestingly, about 38 unique genetic alterations in these trials were identified for enrollment, meaning there was an experimental or known drug to target these particular abnormalities. In terms of identifying new actionable targets, Ross and colleagues published just recently in JAMA Oncology a study looking at carcinoma of unknown primary. So in this study, their objective was to discover opportunities for targeted therapies in patients with carcinoma of unknown primary that are not currently searched for in routine practice. So they studied 200 patients, and they found abnormalities in 217 genes, again, less than that 300 that we talked about earlier, 30 of which were clinically relevant. Interestingly and importantly, and one of the concepts I made earlier, and which we will come back to, is that almost all patient samples had at least one clinically relevant genomic abnormality, and the mean number of abnormalities was 4. It wasn't just a single in most cancers, but multiple. So, is this approach that we've been describing feasible? And so, again, a recent publication, EPUB ahead of print by Merrick Bernstam and colleagues, entitled The Feasibility of Large-Scale Genomic Testing to Facilitate Enrollment onto Genomically Matched Clinical Trials. In the feasibility study, which has now been published electronically, an electronic version, 2,000 patients with advanced cancer were studied. There was broad-based genomic testing. The results showed that 35 of the genes were identified were currently actionable based upon known approved or experimental drugs. 39% of patients had at least one mutation in one of these genes. And unfortunately speaking to, at this moment in time, perhaps the lack of feasibility of this approach is that only 11% of patients were enrolled in genotype-matched trials targeting these alterations. And that isn't necessarily a bad thing, even though this study showed a relative lack of feasibility to this approach. And the authors identified a number of reasons about why it may not be currently feasible, but how that can be addressed going forward as more drugs are available, more clinical trials are available, more drugs are found to be, more targets are identified, more drugs are made to target those abnormalities. So the current limited feasibility based upon this study is likely to change as we move forward, and it may become increasingly feasible. But at the moment, in this moment in time, it's, the feasibility is limited. So to address this, there's probably at least 5 novel designs that are being proposed to be able to move this process of precision medicine forward. The first are what are called basket trials. And in a basket trial, test treatments are based on mechanism of actions as opposed to an organ or histologically defined tumor. Secondly, there are now umbrella trials in which you test multiple drugs for a single disease. Very interesting and very complex would be adaptive designs, where treatment is matched to patients during the conduct of the study based upon responses as new information becomes available, new drugs become available, and there are a few of these, as with all these, there are several examples of all these now becoming operational. I've lumped together registries and databases, appropriately or inappropriately. These may be completely separate, but these are tied to the precision medicine initiative that President Obama has proposed. And these registries and databases will allow for in silico research, if you will, so the ability to really analyze massive amounts of clinically relevant information in clinically annotated tumor samples where the biology is being defined. And then finally, I put up here N of 1 clinical trial. So this is sort of the holy grail of personalized medicine that a lot of people espouse, which is that every individual gets a therapy which is truly tailored just to them. That's probably an unrealistic expectation at this moment in time. Is probably a very worthy goal to work towards. And the approaches that are being suggested for N of 1 clinical trials will require massive amounts of data on the individual about their tumor, about their host gene polymorphisms, about their environment. So for every single individual, the collection of massive amounts of data that would be used to try to do N of 1 clinical trials. So taking one of these designs, basket design, asking the question, are basket designs feasible? So Lopez-Chavez and colleagues just recently published what I think is the first completed and published basket design trial for cancer. This was known as the custom study. And in this, they studied 647 patients with advanced thoracic malignancies. 88% of these tumors assessed had at least one gene abnormality. And when they looked at these abnormalities and the ability to respond to a targeted drug, in the EGFR mutation patients who had non-small cell lung cancer, the abnormality rate for that particular abnormality was 22%, and 60% of patients responded to erlotinib, or a drug targeting EGFR, which is a very, very nice result. The second abnormality was KRAS, also in patients with non-small cell lung cancer. The mutation frequency rate was 24.9%, and unfortunately, the drug targeting KRAS did not reach its primary endpoint, and the response rate was only 11%. Importantly, the completion of accrual to all the other arms of the study was shown to be not feasible, again suggesting that at this moment in time, based upon the targets, the number of actionable targets, and the number of approved or experimental medications that are available, and patients' willingness to participate on the studies, etc., the feasibility is limited at this moment in time. Again, the authors suggested ways that this study design could be improved. And of importance, one of the ways that this could be improved would have been using an adaptive design where new drugs and new arms could have been added throughout the course of the therapy that may have improved the feasibility of the approach. So specifically, ROS1, RET fusions, etc., had they had an adaptive design, may have facilitated the success of this particular study. Next. There are a couple of ongoing and high-profile studies with these novel designs facilitated through the National Cancer Institute, and I would highlight just two of these. And I should add that there are other similar studies that are being done by other organizations such as ASCO and other groups, but just two from the National Cancer Institute. So the NCI MATCH study, which will be operationalized through ECOG and Akron, will enroll 3,000 patients with solid tumors and lymphoma. 200 genes will be analyzed, and there have been commitments for 40 drugs from 20 pharmaceutical companies to help support this particular study. And so this study is now recruiting patients. Secondly, the LungMap study, also through the NCI. This is a phase 2-3 clinical trial design. Patients with advanced squamous cell lung cancer. Patients will have genetic and genomic analysis, and then the results will pair patients to targeted therapies based upon these particular results. And again, this study is actively recruiting patients. So what is fueling this recent progress in precision medicine? So I think there's a number of factors. First, the biology. The advances that have been made in biology over the past several decades are just extraordinary. And what we're learning, as I said earlier, is the biology is exceedingly complex and exceedingly beautiful. And so it's a remarkable time to be in science and in medicine as a result of these enormous advances in biology and our understanding of the host and the tumors. And again, the focus on genomics within the precision medicine world, this has been made possible because of high-throughput screening, deep sequencing, et cetera. And just a few years ago, it took weeks to sequence the genome. The initial Human Genome Project took years to sequence the genome. And now with modern technologies, you can sequence multiple genomes in a single day based upon the capability of any individual laboratory. There's also now the ability to store massive amounts of data, which these initiatives and approaches will require. And so if you look back just a short period of time ago, in 1985, to store 1 gigabyte of data cost about $71,000. Today, 1 gigabyte of data costs about 3 cents. And so the ability to store massive amounts of data greatly facilitates these approaches. And then our ability, as a result of bioinformatics and our advances there, the ability to analyze large data sets gives us the ability to better model disease, understand the biology, and potentially match best therapy based upon biology. So what are some of the challenges that physicians, clinical investigators, biotech and pharma, regulatory decision makers, et cetera, will face with this approach? So, as I mentioned earlier, currently there are a limited number of pathways that are identified that are relevant for cancer, a limited number of genes, and a limited number of actionable genes. The focus on the genome is probably overly simplistic because, like a lot of biology in our work, that's working within a single silo, if you will. And so our ability to work across multiple platforms and model diseases and understand diseases across multiple platforms probably could be necessary to really make significant progress. So by that, I mean we'll have the genome, which is terribly important, but we also need to understand the proteomic silo, if you will, the metabolomic, kineomics, epigenetics, the host, et cetera, et cetera, et cetera. So being able to model these diseases across platforms is a current challenge, but the opportunity there is significant and profound as we better understand how to cross-model disease. And so as we can now create maps for what's wrong, what's abnormal in any individual's cancer cells, we can see that the number of potential targets is enormous, and how to make those actionable is going to be a challenge, but also a great opportunity for improvement. So that said, we have a profound number of potential targets. We then, as we understand these targets, which ones are important, which one of these many, many abnormalities in a cancer cell, which can be one, multiple, dozens, and in some cancers, hundreds of abnormalities in any individual cancer cell, which one of these are important, which are drivers, central mediators of cancergenesis, and which are just along for the ride. And if you target them, it doesn't matter. How many targets do you have to hit that are actionable? As we'll talk about in a moment, one of the abiding lessons of cancer we've learned over the past 60 years is there's very few cancers where you can treat it with a single drug or single therapy. Cancer is exceedingly complex. There's multiple redundant pathways, and so it may be necessary that we have to hit multiple targets. How do we prioritize the drugs and based upon the number of targets that we have, and then the availability of clinical trials to be able to ask and answer all these important questions. So coming back to this theme that it's highly unlikely that for most cancers a single drug targeting a single abnormality is going to be successful, and it's very likely that these new targeted agents are going to have to be used in combination with conventional therapies, conventional chemotherapy, surgery, radiation, blood and marrow transplantation to be effective. And so as we add these new agents onto conventional platforms of cancer therapy or in combination with other targeted agents, defining the safety and efficacy of those combinations will likely require fairly traditional phase 1, phase 2, and phase 3 studies to prove efficacy and safety of those combinations of approaches. Coming back to one of the central premises of precision medicine is will it have the ability to reduce healthcare costs? And that's an important question. We know that just the diagnostic approach, the genetic testing, the companion diagnostics, is likely to increase, and one current estimate is that by 2021, $15 to $25 billion will be spent on genetic and molecular testing of patients. There also is the question of equity because of the expense and access to testing and the cost of new drug development. Will the testing and will the therapies only be available to the wealthy and to the rich? And so is there going to be equity with these type of approaches? Next. And then there are a number of implications about knowing someone's genomic sequence. And so it's beyond the scope of today's webinar, but just as a reminder, there are enormous ethical, legal, financial, and social issues associated with knowing and storing an individual's genome. And then finally, in terms of challenges, while these approaches in medicine will continue to be increasingly precise and increasingly personalized, are N-of-1 trials truly realistic and safe and effective and informative? And the answer to that is perhaps, but it's probably not in the near future when we get down to a single individual patient. So as a result of this enormous complexity in biology and data management and clinical trial design, one question will be what kind of CROs will be important to pharma and biotech and other parts of the drug and therapy development sectors to be able to execute these complex trials. So CROs that are full service with the ability to integrate all of these required elements will likely be necessary to conduct these complex studies. CROs that are physician-led, where the medical and scientific expertise to understand the biology, the targets, the clinical trial designs, and ideally CROs that are able to recruit disease-specific key opinion leaders to work in those CROs who truly have a deep understanding of these diseases and biology. Statistics and data management, especially with these novel designs, is going to be critical, as is IT infrastructure to be able to store and analyze big data. And finally, CROs that have significant regulatory expertise because of the complexity of these studies. So in summary and in conclusion, I would suggest that the biology of understanding cancer and these diseases is advancing at a truly remarkable pace right now, and that improvements in the outcome of patients is undoubtedly going to follow this new understanding. The precision medicine approach, or the increasingly precise medicine approach, as I would advocate we use, is far more complex than most understand, and based upon some of the more just recently published studies about feasibility for this approach is still being assessed at this moment in time, and efficacy and safety are yet to be defined for these approaches, since feasibility is still really the major question. And current results would suggest that these approaches are in their infancy. Feasibility is even limited based upon the current data, but that gives us a baseline to begin the advancements forward. And this is still experimental, and in the end will require partnership, complementation with more traditionally defined phase 1, 2, and 3 studies to truly study safety and efficacy of these approaches. And my final comment was that despite the great excitement in this area and great opportunity in these approaches, it's not quite ready for prime time, meaning don't try this at home yet. And by not trying this at home, I mean this probably should not be done outside the context of a prospective clinical trial, since the safety and efficacy of this approach has not yet been proven. So Morgan, that concludes my presentation, and I will turn it back over to you.
Great. Thank you so much, Dr. Smith. That was an excellent presentation, and you raised a lot of great questions. We'll try to get to as many of those questions as possible, so I will get right to it. Let's see. What role do you anticipate genomics, pharmacogenomics, epigenetics, transcriptomics, proteomics, and metabolomics will play in the development of precision medicine? And do you think that the current tools and technology are available to provide an integrative, personal, quote, omics profile to facilitate the success of precision medicine?
Thank you, Morgan. That's an outstanding question. I touched on it briefly, but to expand upon it to answer that particular question, I think for the near future, current and near future, we're continuing to work within these silos, if you will, and you just, as part of the question, those silos were identified. And so I think we're going to learn a huge amount about these approaches by working within these silos. So that's not necessarily a bad thing. But I think to understand cancer better, we're going to have to better understand the cancer through cross-platform modeling of disease. And there's a number of academic and industry partners that are trying to do that right now. The bioinformatics capability is quite extraordinary, also in its infancy, but making rapid progress to be able to do cross-platform modeling. So I think ultimately, as we move to truly personalized and highly precise treatments, they're going to require cross-platform modeling. So at this moment in time, I don't know that we either have the technology or understanding of how to do that terribly effectively, to take all these various platforms into account to truly individualize therapy. But I think that's the direction that the field is heading. But again, it's in its early days, but making progress.
Great, thanks. I will move right along here. An emphasis on pharmacogenomics seems oversimplified given the number of compensatory responses associated with the modulation of any given targeted gene or pathway. Furthermore, the current focus on actionable targets only represents a limited number of genes or pathways. How will precision medicine overcome the complex and unique biology associated with each individual and tumor?
Thank you, Morgan. Also a very good question, which I touched on briefly. So to reiterate one of the points I made earlier and to expand a little bit upon this. So again, as the questioner has rightly pointed out, cancer is exceedingly complex, and as a result of the evolution of cancer, there is rarely a single pathway that the survival of that cancer is dependent upon. And so if you hit one important actionable driver, it's very likely that the survival pathways in that cancer will simply signal around that particular pathway to a different survival pathway, and also the ability to develop resistance to therapies, including these targeted agents, where we now know that resistance does develop as a result of even targeting very central drivers of cancer. So the holy grail of cancer, where you could give a patient a single drug, is probably unrealistic in most diseases, and so it's probably going to have to be combined with either other highly targeted drugs. And again, there, if you're putting new novel combinations together, that would be inappropriate and probably dangerous until phase 1 safety and efficacy studies are done in phase 1, phase 2, fairly traditional settings. Also, you're going to have to pair these drugs with conventional therapies, chemotherapy, surgery, radiation, and transplantation. And again, until you prove that they're safe and effective in phase 1 and phase 2 studies, just doing it for the treatment of an individual patient without data to show safety and efficacy would be inappropriate at this point. So I think we're going to learn a lot by targeting therapies to specific abnormalities. There's likely clinical benefit to be derived from that. But I think ultimately it's going to be combinations of either multiple targets or new targeted therapies in combination with conventional therapies. The other concept here, which I didn't mention earlier, which may be relevant, is that everything we've talked about today has been further defining unique abnormalities in an individual patient cancer. We haven't talked a lot about the host and the host factors involved in that and how drugs are handled and metabolized and pharmacokinetics. And we haven't talked much about pharmacodynamics at all either. But we've been splitting. And a lot of this precision medicine is splitting down based upon individual abnormalities and targeting those very specific abnormalities, of which there are multiple in cancer. Another approach, which is terribly exciting, is not based upon the splitting phenomena, where the number of patients available in any small specific group become increasingly limited, but can we relump? Can we put patients together in large groups, develop a therapy which may be effective that could kill cancer cells regardless of all the complexity of what's happening inside that particular cancer cell. So if you ignore all those dozens and hundreds of abnormalities in the cell, are there pathways to target it that are independent of all those abnormalities? And so there are a number of approaches that are being developed to relump, if you will. And so a number of cellular therapies are doing that. CAR T cells, for example. So when the work by the group at Carl June and Stephen Grupp and David Porter at the University of Pennsylvania, among others, when they use this CAR T cell therapy in children who have acute lymphoblastic leukemia, they're targeting CD19. What's happening inside that leukemia cell becomes much less relevant because the immunotherapy approach is targeting something that lumps everything together, if you will. New targets like CD47, the do not eat me signal on macrophages. If there's a way to target that, it relumps. It doesn't matter necessarily what's happening inside the cancer cell if you can find a lethal way to target broader pathways. So I think precision medicine, another thought process that people should be aware of, is it isn't dependent necessarily upon continuing to split into very, very small groups of molecularly defined patients. Another approach is to find ways to relump them into sort of central lethal pathways and therapies that would get around this splitting process. So, Morgan, next question.
Wonderful, thank you. Next question. The emphasis of precision medicine appears to be on the biology of the tumor. Is it reasonable to expect that this is a bit naive and that the biology of the individual could be just as important in the development of precision medicine? And if so, how would you successfully interface this information in the development of precision medicine?
Thank you, Morgan. Again, an excellent question. And I think this highlights at least two additional features, one of which I talked about earlier. So the host is exceedingly important and how the host is going to handle any individual therapy. And so the pathways we've been talking about thus far have been targeted to the cancer itself and not to the host. And so I think one of the lessons that we're learning now is considering the host and how they're going to handle the therapy is equally important. It ties in first to the concept I talked about earlier about cross-platform modeling. So one of those models are host gene polymorphisms and how to better integrate those into disease models, if you will, so that the host is taken into account. And again, there is daily new information around the host that's going to have to be tied into these broader models and have to be considered as we develop the therapies. The other, as it illustrates precision medicine, there was recently beautiful work presented by Smita Bhatia and her group within the Children's Oncology Group, where she looked again at a very old drug that we've used in pediatric ALL, acute lymphoblastic leukemia, since the 1940s, 1950s, the thiopurines. And what Dr. Bhatia and her colleagues asked was, among children who take their medication, so they're adherers, so you don't have the issues with did they take the medicine or not, of those who take their medication, if physicians are constantly changing the dose of that particular medication based upon the patient's white count as it goes up and down or various other parameters, inter-patient variability, if the patient's levels of the thiopurines go up and down constantly, those patients have a worse outcome. So this suggests opportunities for further studies where we can take a drug which has been around since the 1940s, and we're still learning how to use that based upon how an individual patient metabolizes that drug and the complexity of social situations where how do you have children be adherent, patients adherent to the drug, and physician behavior about should we be changing the doses based upon parameters that may or may not, at the end of the day, be important. So a lot of opportunities still with old drugs to personalize it and make it more precise that could improve the outcomes. So understanding the host is terribly important. Next question, Morgan.
Thank you. If precision medicine is directed at specialized patient treatment, would you anticipate this to increase healthcare costs? And if so, how will this be supported in the current healthcare system?
I can make an attempt to answer the first part of that. The second part, I don't know that I have a good answer for, but I think it's a very good and very insightful question. So again, one of the core premises of precision medicine approaches is that it will result in healthcare costs. And if this is actualized, I think it has the distinct possibility to take cost of healthcare down because you more effectively treat the patient. It's cheaper to live than it is to die. It's better to be able to treat the patient, minimize complications, keep them out of the hospital, not manage all the complications, not have to use multiple cycles of therapy for when they relapse. So if the approach is actualized, I think there's a very distinct and real possibility it could drive down healthcare costs. That said, this will have to be offset in the calculations by healthcare economists, which I'm not one. So everything I'm saying is with a bit of naivete. But I showed you the cost of doing the testing. And there's going to be an enormous increase in the cost of actually doing, and this is likely to get even more costly as we will increasingly have the ability to test more cross-platform model diseases-based therapies, these targeted therapies upon multiple factors. And so the testing likely would increase. So where the difference is between the cost of doing this, the cost of developing new drugs, the cost of drugs that are ultimately approved, versus having better outcomes and reductions in cost of care because you're effectively treating patients. I think the healthcare economists will have to do those calculations. But I think one of the great promises of increasingly precise medicine is it has the very real potential to reduce costs if we can better treat patients and improve outcomes.
Great, thank you. Next question. Given the recent advances in technology, what do you see as the current rate-limiting step in achieving precision medicine?
Also an excellent question. I think that if you look at some of the challenges and the bottlenecks right now, as I talked about earlier, we have a limited number of genetic abnormalities and a limited number of targeted drugs, either approved or experimental right now. That said, I think those are manageable bottlenecks at the moment because the genes that are abnormal, many of these actionable targets for which we have drugs are central pathways, and many of those are drivers of malignancy. So I think we have a reasonable compendium of agents, both approved and in the clinical trials process now or in the immediate future, to begin to ask the initial feasibility and safety and efficacy, look for efficacy signals here. So I think the real bottleneck, to answer the question at this moment in time, is having a sufficient number of clinical trials available that can capture these groups of patients. So the NCI studies we talked about, the American Society of Clinical Oncology study, potential studies by the Leukemia and Lymphoma Society, many academic groups that are doing this, many biotech and pharma groups that are doing this. I think the more clinical trials we have available that would be available for investigators and patients to enroll on will help us to answer the initial central questions around feasibility, safety, and to begin to get efficacy signals. So availability of clinical trials, I think would be my answer to the question, Morgan.
Wonderful. Thank you. Depending on how much time we have left, we might be coming to an end of our questions here, but we'll try to get a couple more in. You reference approximately 300 genes that may be involved in cancer. Is that based on gene products initiating cancerous growth? And what about epigenetic phenomena above and beyond gene products? Have epigenetic mechanisms been identified with the onset of cancer?
Excellent question. So the genes, those 300 genes, are typically genes, and there are a number of references which can be included to support that at this moment in time, and the mapping that's been done to try to define the number of sets. So these are typically mutations, but also does include other abnormalities. Epigenetics is increasing, our understanding is that it's increasingly important to the biology, and there are currently clinical trials targeting epigenetic pathways. And again, this would be another platform we have to integrate in. And so it's not just the code of life, it's just not the map that you're given. I think this is where the person who asked this question is heading to, and our ability to target genetic abnormalities and the products of those genetic abnormalities. But how does this incredibly complex field of epigenetics bear on that, which genes are turned on and which are turned off, and both within the cancer and within the host. And so there are initial approaches with epigenetics, but again, those would be in their very early initial stages as well. Very interesting, very exciting, but we're early in that process.
Great. I think we have time for one more question. This will be our final question, and we've had so many great inquiries today, and I'm sorry we couldn't get to them all. Last question, to what extent will immunomodulation and targeted manipulation of the immune system therapeutically add to the effectiveness of therapies directed at actionable genomic targets?
Wonderful question. I love that question. So if there's any topic that's hotter than precision medicine right now, it's immuno-oncology. And again, this is stimulated by a number of very, very wonderful and exciting approaches with cellular therapy. I referenced Carl June and his work with CAR T cells as one example of that. Checkpoint inhibitors and ways to manipulate the immune system, PD-1 and PD-1 ligand approaches. So I think that these are going to be very, very important processes. And coming back to my prior comment to one of the questions about splitting versus lumping, I think one of the great benefits of this approach, this immunomodulation and targeting, comes back into perhaps the relumping process. How do we find lethal ways to kill tumor cells that are not necessarily dependent upon all the complexity of what's happening inside the cell? And as we know, the immune system is extraordinarily powerful if we can find its ways to harness it. And as a part of that, of the central driving principles of cancer and why people have cancer, one of those is that cancer is exceedingly effective in evading the immune system. And so for the science nerds out there, the analogy that I use is Star Trek, so that the Romulan warbird is sitting right next to the Enterprise, but the Enterprise can't see it because it's cloaked. And cancer cells are cloaked. The immune system just can't see it. So a lot of this very, very exciting work has great promises that they uncloak the cancer cells. And so they become visible to the immune system. They're no longer hiding in plain sight. And so I think there's great, great opportunity here. But again, these are going to, they have a number of challenges. They're going to require some probably different clinical trial designs with different levels of complexity. But it's a very, very promising and exciting area.
Wonderful. Thank you so much. It's time to wrap it up. I'd like to thank everyone for attending this Fierce Live webinar and for submitting so many great questions. I'd especially like to thank Dr. Smith for participating and MedPace for presenting today's webinar. The webinar was recorded, and you'll be able to access the recording within 24 hours on the same page that you used to register for the event. So thank you again, everyone, for joining, and we look forward to seeing you at future events.